Hello, how are you? Martin Mlondo is my name. Actually, a medical worker. Right about today, I want us to talk about blood. And this is actually among one of the subunits of anatomy and physiology. And I must appreciate that blood is actually under connecting tissues, and you must appreciate that connecting tissues are also a sub actually division in anatomy. And without actually wasting time, right about now, I want us to start and talk about blood and its composition. When someone talks about blood, what does blood mean? Blood is actually a specialized fluid. It's a specialized fluid. Are we together? It is a specialized fluid connecting tissue that contains of cells that are suspended in a fluid matrix. Can I come again? I said blood is a specialized fluid connecting tissue that contains of cells that are suspended in a fluid matrix. And I must appreciate that blood actually is maintained actually at a constant flow through the body by the help of what we call a pump, which is known as the heart. Are we together? So the heart is one of the pump in the body that maintains a constant flow of blood. And we must also know that it is actually a complex substance that is actually composed of a lot of cellular elements that is with blood. So allow me right away look at the functions of blood. What are some of the functions of blood? We must appreciate that blood transports oxygen from the lungs to the tissues, and carbon dioxide from the tissues to the lungs. That is for excretion. And still, we must appreciate that also blood helps in transportation of nutrients via the alimentary canal. Are we together? That is through absorption. Are we together? So, still, blood acts as a messenger function which actually helps in the transportation of different hormones that are secreted by the endocrine glands. Still, blood helps in actually maintaining the temperature of the body. That is actually helping in homeostasis. So, we must also appreciate that blood actually helps actually protects the substances, actually helps naturally production of different protective substances like the antibodies or what we call the immunoglobulins. Still, furthermore, we must appreciate that blood helps actually in the clotting factor, which actually helps in actually the body's defense repair mechanism. Are we together? Still, blood also does what we call an hydraulic function. That is, we can see that actually during erection of the penis. That is actually what we call also, and as we look at the clitoris, it's also resulting into what we call erection. It does what we call a hydraulic function. So there are some of the functions of blood. I can actually summarize them down. I talked about transportation of oxygen, that is from the lungs to the tissues, from also carbon dioxide from the tissues to the lungs. That is helping in gaseous exchange. It also does what we call transportation of nutrients through absorption, that is through the alimentary canal, acts as a messenger function. It also helps in the height regulation or temperature regulation of the body, helps in the production of antibodies, helps in clotting, helps in actually maintaining the body pH, and also actually helps in what we call the hydraulic function. Alright, about now, allow me proceed and I talk about the composition of blood. And you must have seen that blood is composed of plasma and actually, which is actually a clear stray. That is what we call a pale yellow fluid. You must appreciate that plasma actually we must appreciate that plasma is a cellular substance found in blood, which is actually constituting about 55% of blood. And different types of cells are suspended in it, consisting of about 45% of blood volume. What do we mean? You must appreciate that blood is also composed of different cells in it. You must appreciate that under blood we have a lot of cells like the red blood cells, the T cells, the B cells, and so many other cells. Yeah, so if I could actually pose a question, how many types of cells are composed in a human body? That is actually a question for you to answer in the comments to the discussion. If I further proceed, you must appreciate that blood consists of majorly two components. That is the plasma and the cellular part. And actually, these two components that are composed of blood, you realize that plasma is less dense than the other part of blood, which is containing of the cells and the hemoglobin. You must appreciate that when we carry out centrifugation, or what we call spinning under gravity in the laboratory, and we put a sample of blood in a test tube, after spinning it, we shall see the permeable substance rising on top of blood. So that means plasma is less dense than the cellular composition. So having discussed that, you must appreciate that the cells are always heavier than the plasma, so they always sink. They always sink below at any sample of blood. So you must also know that a normal person has 5.6 liters of blood, and the normal person, that is a normal person we are talking of, is that person who is actually approximately to 70 kilograms. So when you are less than 70, you are not normal. Above 70, you are not normal. So we believe that a normal person should have 70 kilograms, and therefore he has 5.6 liters of blood. So if I further proceed and I actually talk about this concept of how much blood a person has in the body, for any person who is actually below 70 or less than 70 kilograms, and they actually tell you there is an objective which actually is usually set. They can say what is the normal range what is the actually, they usually say what is the total or what is the amount of blood in a normal person, and they can actually give an objective: A 5, B 7, C 4, D none of the above. We shall go with the 5, though it is not 5.6 as the exact value, but it is near to the answer. Are we together with what I'm trying to explain to you? Allow me proceed and actually look at another concept, and I tell you that ladies have got less actually blood compared to males, and this is due to the fact that they go through menstrual seasons and also have a period where they go, where actually during childbirth, that is the pregnancy level, where they lose a lot of blood. Therefore, ladies have got less blood compared to actually males. So, if I proceed, having actually given a brief clue about what blood is, allow me now look at plasma as a component of blood. And you must appreciate that plasma is actually made up of water, and the water in it is actually ranging between 90 to 92%. And with actually, you must appreciate that with substances dissolved in it. So, what are those substances that are dissolved in plasma? So, we are looking now at the composition of plasma. We have what we call the plasma proteins. We have what we call the electrolytes, or what we call the inorganic salts. We have what we call the nutrients, mainly from digested foods. We also have what we call the waste products. We also have what we call the hormones. We also have what we call the different gases, that is the oxygen, carbon dioxide, nitrogen, enzymes, plasma, proteins, and very many other concepts. Let us agree and also appreciate that plasma proteins make up around 7% composition in blood. So we must also appreciate that they normally retained within blood. The plasma proteins are mainly retained within blood, and they are normally produced by what we call the liver, as we shall see in the coming as we go on. Allow me tell you that blood having plasma proteins is very important. How? These plasma proteins help in actually maintaining the actual or creating an osmotic pressure that actually helps to keep fluids intact. Are you together? So without the plasma proteins in the body, we would have seen water. That water actually being actually occupying the interstitial spaces. So a person who actually has water in the tissue spaces always has a condition called edema. Are you together? So plasma proteins, except the antibodies, are all found in the liver. And I must appreciate that the PDF I'm always referring to about blood actually was made by my tutor, that is Mr. Ambrose Biamkama. So we are so grateful for this. So allow me talk about some of the examples of the immunoglobulins as examples of plasma proteins. Remember, I told you we are looking at plasma as a composition of blood. Then I told you under plasma, it is actually made up of water. That is 90 to 92%. But the rest part is made up of other components. That is the enzymes, gases, hormones, waste products, plasma proteins, electrolytes, and nutrients. So we are looking at plasma protein as a component of plasma. So now we talked of plasma proteins. I talked of actually plasma proteins covering 70% of plasma, and we said they are mainly retained in blood, and they are responsible for maintaining osmotic pressure that actually prevents edema by actually keeping fluids intact in their space. They also help in actually production of different antibodies in the body. So now allow me to look at some of the examples of the antibodies that are found within the liver. So these antibodies include the albumins. The albumins are actually formed from the liver, and they are most abundant plasma proteins, about 60% total. Their major function is to maintain a normal plasma osmotic pressure. They also act as carrier molecules for actually free fatty acids and some drugs and some steroids. What do we mean with carrier molecules? You realize that you find that some of the fatty acids have got large particles. So this ones help them to actually cross over. They go and carry out their function in the body. So you must always note that less of the albumin in the body leads to less albumin in the body, or loss of albumin in the body leads to what we call edema, because there will be a lot of accumulation of fluids in the body tissues. The other example of antibodies, what we call the globins, and these are found in the liver, and some are found in the lymphoid tissue. So the lymphoid tissue, you must know that is an example of a connecting tissue. What are some of the functions of the globins? They also act as antibodies. With this, you must appreciate that antibodies are very important in fighting microbes in the body. So they also help in transportation of hormones and mineral salts. Ig actually, there is a hormone called thymosin or thyroid. So it is actually a hormone found also in the body. The other function of these globins actually, they also actually help in actually what we call inhibition of different enzymes. That is the alpha-2 macrobins which inhibit the trypsin activity. So with the blood, you must enjoy this topic. It is a very simple topic. Allow me proceed, and I tell you that we also have what we call clotting factors, that are some of the examples of compositions of plasma, and these clotting factors include the fibrinogen, which helps in actually the clotting action. So let us look at another component of plasma, having seen plasma proteins. let us also look at what we call the electrolytes or inorganic salts, and these actually include the calciums, the potassiums, the nitrates, and so many other things. And we must appreciate that an electrolyte is an ionic compound that which when dissolved in water forms electricity, and that electricity has got different functions in the body. So what are some of the functions of electrolytes in the body? They help in what we call the muscle function in the body. They also help in actually maintaining the pH. They also help in and actually maintaining that the different fluids are kept in their portions. If I proceed, allow me look at the different nutrients that are also found in plasma. We have glucose, sodium, fatty acids, vitamins, and so many other things. What are some of the antibodies that are actually produced what are some of the waste products? Because the antibodies, we have already seen that we have the albumin, the globins, we have the albumin, the globins, and so many other things that are produced within the liver. So, what are some of the waste products that are produced by the plasma? We have what we call the urea, or what we call the carotene. It is a mineral which is actually produced by plasma. We have what we call urea and carotene, and these are waste products of what we call protein metabolism. So as protein metabolism takes place, you must appreciate that urea is always given off, and it is very, very important for our bodies to lose urea. That too much urea concentration in the body will always lead to different problems. And we also have carbon dioxide, which is actually released off in the body as a byproduct of excretion. So those are some of the waste products that are actually lost within actually blood. So what are some of the hormones that are actually found within blood? So we are talking about the compositions of plasma. Are we together? So with the hormones, you must appreciate that they are synthesized by the endocrine gland. So blood transports these hormones to their targeting cells and organs. Some of the gases include that are actually found in plasma. We have oxygen, carbon dioxide, nitrogen, which are all dissolved in plasma, and oxygen and carbohydrates are also transported in combination with what we call red blood cells. Having looked at the first composition of blood, which is plasma, allow me proceed to the last composition of blood, which is called the cellular part. And the cellular part, we shall majorly look at three types of blood cells. That is the erythrocytes, or what we call the red blood cells, the leukocytes, or what we call the red blood cells, the thrombocytes, or what we call the platelets. And I must appreciate that blood cells are actually synthesized mainly in the red bone marrow. I told you the bone marrow is a tissue under the lymphatic system that helps in the production of lymphocytes under the lymphatic system, but in addition it also produces for us what we call cells, or what we call the body cells. And these body cells are very important in the body, and you must appreciate that they always and actually in the bone marrow we shall see all blood cells originating from what we call the pluripotent stem cells. And with this we mean that cells are capable of actually developing into one or a number of different cells. So excuse me. So after they go through the several development of stages, they actually enter blood, and allow me discuss more about the different what we call cell formation, which is called hemolysis. And it is called hemolysis, and with actually with hemopoiesis. I'm sorry for that. The process by which different cells are produced within the body is what we call hemopoiesis. So allow me look at the different types of cell formation, or the different hemopoieses of different cells. And right about now, I'll start with what we call the red blood cells. And you must appreciate that the red blood cells are the most abundant type of blood cells in the body, and actually they are produced through a process called erythropoiesis. Erythropoiesis, which erythropoiesis falls under hemopoiesis. So these concepts here are quite challenging, so you just have to be very careful. You must appreciate that red blood cells actually cover the biggest part of the cells in the body, and they make up around 99% of the body cells. They are made up of biconcave discs with no nucleus. They have a diameter of probably 7 micrometers, and their major function in actually is to transport oxygen. They also carry some carbon dioxide, and their characteristic shape is what we call a biconcave shape, which is situated to its function. There is a reason as to why it is made up of a biconcave shape, and it is advantageous. So the biconcave shape actually increases the surface area for gaseous exchange. Remember, a biconcave shape is always thin in between here, so it always increases the surface area through which actually gases are exchanged in red blood cells, and the thinness of the central portion enables that. So these cells are flexible, so they can always squeeze and actually pass through the narrow capillaries, so they contain no intercellular hemoglobin. So when I talk of hemoglobin is actually a large pigmented protein responsible for gas transport. And you must appreciate that red blood cells actually in the body lose their nucleus as they mature, thereby becoming incapable of division. So what do we mean? That red blood cells, once they actually produced, they lose a nucleus. So they always live for a particular lifespan, that is for 120 days, and after 120 days, they will always die. Are we together? So, allow me, you will have to see a diagram there that shows the different processes through which hemolysis, hemopoiesis takes place. Yeah, hemopoiesis takes place. It shows the first line there, shows the process by which the red blood cells are produced, that is through erythropoiesis, and we shall see that it goes through the first stage called the proethoblasts, which actually transmitted into the ethroblasts, which ethroblasts become reticulocytes, which reticulocytes become mature erythrocytes in the long run. That is actually a diagram that shows a pluripotent stem cell having being on top. It is branched into different cells. So the first line shows the production of red blood cells through erythropoiesis. The second one shows actually shows the platelets, how they are produced. The other one shows how the white blood cells are produced and how they actually are specialized. So right about now, allow me to tell you that red blood cells actually create more space for hemoglobin, for hemoglobin, actually, and this helps red blood cells. Them actually creating more space for hemoglobin helps in high transportation of oxygen in the body. And red blood cells are produced in the red bone marrow, in the ends of the long bones, in the flat bones, or what we call irregular bones. What are some of the adaptations of red blood cells to their function? They have got a thin membrane in the center to allow easy entry and exit of different gases, that is the oxygen and the carbon dioxide. They also have a biconcave shape to increase surface area for gaseous exchange. They are also flexible, that can actually help them squeeze and pass through capillaries. They also actually lack intercellular organelles. This actually leaves more room for a hemoglobin for hemoglobin concentration, which actually increases oxygen concentration in the body. And so, allow me look at the breakdown of the process called erythropoiesis. And erythropoiesis is the process of development of different red blood cells, and it takes 7 days, and it is actually influenced by what we call the erythropoietin hormone, which is produced within the kidneys. So the erythropoiesis begins within the pluripotent stem cells, and these stem cells transformed into what we call the proerythroblasts, which then grow into what we call the erythroblasts. So when someone tells you to explain erythropoiesis, you always start and define the process of development of red blood cells. It takes 7 days. It's a process influenced by what we call the erythropoietin hormone produced within the kidneys. And erythropoiesis begins within the pluripotent stem cells. And these stem cells are formed into what we call the proerythroblasts, which then give a rise to what we call erythroblasts. So after having seen or having got the erythroblasts, during these two stages, you must appreciate that. You must also still appreciate that during these two stages, there is a production of huge numbers of ribosomes, and these ribosomes are the proteins, are those proteins found within the cells. And you must appreciate that under still this process of erythropoiesis, we shall see cells dividing into many hemoglobin synthesis areas. We shall also see ion accumulation which occurs. So we shall also see what we call a color change, which is actually seen in the cytoplasm, where we shall see blue staining ribosomes becoming pink color hemoglobin. So when the cells actually accumulate, the hemoglobin concentration about 35% or actually will always lead to the organelles being actually ejected out. And actually, in so doing, we shall see that the nucleus disintegrates and is pinched off during the cell to collapse inwards, actually causing a bioconcave shape. And after all that, we shall see reticulocytes developing, and reticulocytes are immature red blood cells, which is actually released into blood streams. And when it is in the blood streams, after a period of 3 to 5 days, it will develop into what we call the mature erythrocytes. And all this process can actually increase the speed due to the presence of both vitamin B12 and what we call the folic acids that are actually needed in the red blood synthesis. So, a summary of the process of erythropoiesis. First of all, you must know that it is a process of red blood cell development, takes 7 days. It is influenced by erythropoietin hormone produced by the kidneys. And I also must know that it starts from the pluripotent stem cells. And we shall see the proerythroblasts giving off as the first stage, and we shall see this after the proerythroblasts giving off as the first stage. It will later develop into what we call the erythroblasts. And during the formation of the erythroblasts, we shall see 7 things happening. We shall see ion accumulation. We shall see actually production of ribosomes. We shall see more production of hemoglobin. We shall see color change from blue staining ribosomes to pink. We shall also see loss of organelles. We shall also see loss of the nucleus. We shall also see formation of a bioconcave shape, which later actually results into what we call reticulocytes, which reticulocytes in the long run become erythrocytes after 3 to 5 days when they are in blood. So that is the process of erythropoiesis, and I believe Yeah, that process I told you can be speeded up with the presence of folic acids in the body and vitamin B12. So I believe by now you now understand everything about that. So allow me continue and also tell you that you have to go and look at some diagram showing the maturation of the different erythrocytes in the body. So it needs you to go Google this out and get to know such how the process takes place diagrammatically. All together. So let us look at what we call erythrocyte count as the number of erythrocytes per liter or per cubic milliliter of blood. Number of erythrocytes per cubic liter of blood. The average number of red blood cells count is about 5.5 million cells stroke what we call the millimeters chopped in men. So in ladies, we must know that the normal number of red blood cells is around 4.8 million cells. Yeah, actually, million cells stroke milliliters chopped in women. So we shall see that erythrocyte segmentation rate is the rate at which the red blood cells settle at the bottom of a test tube during centrifugation. And when the erythrocyte when the erythrocyte segmentation rate is low, this actually shows that a person might have actually an infection. So whenever actually they cut out and they see that the concentration of the ERS, or what we call the erythrocyte segmentation rate, is low, it shows that you might have an infection. So allow me to tell you that the ESR is raised when there is a blood disorder like the ploclysmasia. Yeah, so you must also appreciate that the ESR is raised when there is a blood disorder like the it is actually ploclysmasia. So this is an abnormal increased hemoglobin concentration in the body, or during a sickle cell anemia. So measurement of red blood cells is actually done under what we call a process in the laboratory called ESR, which is the erythrocyte segmentation rate. So you must know that I should go and actually read more about the measurement of red blood cells in the laboratory. Otherwise, I will leave it at that, and I proceed and I look at the factors that actually influence erythropoiesis. And the first factor is the presence of proteins. Amino acids are required for synthesis of globin part of hemoglobin, and in starvation in children with kwashiorkor and in protein energy malnutrition, as severe anemia occurs. What do we mean here? That without proteins, your body will actually also have less. We always have less erythrocytes. The other factor is what we call iron. It is essential for red blood cell formation because it is essential in actually formation of hemoglobin molecules. The other factor is what we call the vitamin B12 and the folic acids, or what we call the vitamin. Folic acids are the same as the vitamin B9. So, and these are essential in malnutrition of red blood cells. So, any lack of either of these vitamins may also lead to a deficit or deficiency in red blood cells. Allow me to tell you that we also have another factor called erythropoietin. Erythropoietin, and this is actually a protein which said this is produced from the kidneys. Excuse me. And without this erythropoietin, you will also still have low red blood cells. So we also have other factors that include the low tissue oxygenation. Low tissue oxygenation. And with the low tissue oxygenation, you must appreciate that this stimulates the bone marrow to produce more red blood cells. So whenever we have low tissue oxygenation, we always have increase of production of red blood cells. We also have what we call the endocrine factors. Yeah, that one, go and research on how then you can actually bring out that. So allow me look at the structure of hemoglobin, which is required, and you must know that in blood, iron is actually transported loosely, bound to transport protein called transferrin, and it is stored in the liver. So the normal red blood cell production requires a steady supply of iron. Therefore, you must appreciate that iron is one of the components that makes up hemoglobin. So, and you must appreciate that the hemoglobin is made up of a protein chain part and also what we call an hemo group, or called the iron-containing part. So, go look at that, and you all know, probably you must know that hemoglobin helps in transportation of oxygen, where when it combines together and forms a very unstable compound called hemoglobin, which is actually transported back to the capillaries, hence the association of oxygen. So that is without hemoglobin in the body, we shall not actually have oxygen being transported. Yeah, you must appreciate that the normal hemoglobin levels are 13 to 80 grams per 100 milliliter, that is in male. So, and in females, it is 11.5 to 16 grams per 100 cubic meters of blood. So, probably, I think in the last discussion I did put for you something. It was a document that was showing results of a patient. So I wanted someone to explain to me that the hemoglobin concentration. So it was high. So it was showing that someone actually probably had diabetes mellitus. So I wanted you to go and look at those results and learn how those things actually written. Let us look at the factors that actually increase oxygen concentration from blood. Yeah. What are some of the factors? The first factor is what we call the low oxygen levels, or what we call the hypoxia. Whenever we have low oxygen concentration in blood, we shall always see hemoglobin breaking down to release oxygen. Are we together? So hypoxia is the first factor that leads to high oxygen dissociation. Are we together? So, and hypoxia is the low oxygen concentration. The other factor is what we call the low pH. And we know that metabolically, active cells, e.g., exercising muscles, and leads to releasing of acid, which actually wastes a lot of oxygen. Therefore, need for more under that condition. Still, temperature is another factor that leads to increase in oxygen dissociation. Yeah, and we know that actively metabolizing tissues which actually will actually have high than normal oxygen needs. So during warm areas, you must during warm temperature, we shall see low oxygen used. Therefore, yet during high, we shall see that oxygen association will be much in the body. So allow me try to explain the control of what we call erythropoiesis. And the number of this control of erythropoiesis always relies on the number of red blood cell remains. Actually, we shall see that the red blood cells will always remain fairly constant in the body. And this means that the bone marrow produces erythrocytes at a rate which they are destroyed. All together, as they are destroyed, at the same time they are produced. So this fairly constant state is due to what we call homeostatic negative feedback mechanism. And the hormone that regulates red blood cell production is erythropoietin, produced mainly in the kidneys. And that erythropoietin hormone is very important in the production these red blood cells. Yeah. So with the control of erythropoiesis, you must appreciate that it lies mainly mainly on mainly 5 factors. That is the hemorrhage or low blood volume. It also lies on poor blood flow, anemia. It also lies on an excessive erythrocyte count, or what we call hemolysis, or due to a disease. It is also lying on a reduced oxygen content in the air at high altitude. So, more is actually asked around there, around the control, and it requires you to go and look at a PDF. I'll give to you in our discussion groups. But don't forget to subscribe to all our discussion platforms. On Facebook, we are found at medical discussions by Martin Mulo, and on YouTube, and most importantly, YouTube, go subscribe so that you don't miss anything. We also found on medical discussions by Martin Mulo. So allow me right about now look at the destruction of erythrocytes and the catabolism of hemoglobin. So, hemoglobin catabolism and erythrocyte destruction is also known as hemolysis. Hemolysis is the destruction of red blood cells. It is carried out by what we call the phagocytotic reticuloendothelial cells. Are you together? It seems to be a wider word. We know hemolysis is carried out by what we call. It is actually carried about by what we call a phagocytotic reticuloendothelial cells, and these cells are found in many tissues, and actually their main sites of hemolysis are the spleen, bone marrow, and the liver. So we shall see red blood cells being destroyed much in the spleen, bone marrow, and the liver. And as erythrocytes age, their cell membranes become more fragile and susceptible to hemolysis. So actually, following hemolysis, hemoglobin decomposes into iron, and biliverdin, a green pigment, is produced. So as hemolysis takes place, these are some of the things that take place. We shall see hemoglobin decomposing into iron, and after decomposes into iron, we shall see biliverdin, a green pigment, being what? Formed. Are you together? And this biliverdin is actually stored in the liver. Are you together? Now we have the biliverdin, which is a green pigment. Now, iron is retained in the body and actually refused in the bone marrow to form a new hemoglobin molecule. Are you together? So, now we shall see the globin portion breaking down into amino acids, which actually released into the bone marrow to produce new red blood cells. So, the hemo part is split from the globin part, and it is what we call the biliverdin. Are you together? Now, which is what we call the biliverdin. The biliverdin is converted by the liver into a yellow substance called bilirubin, and this bilirubin is what is secreted as bile pigment. Are you together? And that yellow substance is what we call the jaundice. And the jaundice is a yellow discoloration of body tissues due to accumulation of bile pigment called bilirubin, which is a yellow color. Are we together? So, and you must appreciate that when someone actually develops jaundice, that shows at times it's the sign of malaria. Yeah. So allow me, having discussed for you how red blood cells are destructed in the body, allow me actually tell you that that is what I can talk about the erythropoiesis. Allow me now look at a very important aspect in learning blood, which is the blood groups. And a blood group is a classification of blood based on presence or absence of antigenic substance on the surface of a red blood cell. What do we mean when we talk about that? You must always know that the surface of a red blood cell always carries a different protein called antigen. Are we together? And these antigens are also known as anticoagulants. And these antigens actually react against what we call the agglutins. They always react against what we call the agglutins. So these antigens, which are inherited, determined by the individual's, actually this one determine the individual's blood group. To bring this to your attention, or to make this thing very easy for you, your red blood cell is protected by what we call an antigen. And this antigen actually are the ones that you will give a name of the blood group you hold. Are you together? So if your red blood cells are surrounded by antigens A, are you together? So your blood group A, and you always stimulate an opposite antibody production. That is, you always stimulate the production of anti-B. Are you together? It is the same. If it is B, antigen B on your red blood cells, so your blood group B, so you produce antibody A. Are you together? Are you together? So you must always get to know and appreciate that this part of blood groups is very easy if you pay attention and actually understand it under normal life. So allow me explain to you some of the different concepts of blood groups under this part, and you must appreciate that mainly here we look at what we call the ABO system. We look at the ABO system, and I must appreciate that under the ABO system, it shows about 55% of the population of that has either the following antigens on their surfaces. So the above, which is the ABO system, shows the different blood groups of people. As I've told you, majorly we have 4 types of blood groups. That is AB, and O, and they always actually give actually an opposite ant. So if you blood group A, you give an opposite antigen, actually antibody B. So if your blood group B, ant A. Are you together? So this is actually well understood better when you look at the PDF. I'll send you. And you look at in our WhatsApp group. It shows a table that summarizes down on how we see these people. We shall see these people with these different blood groups being compatible as a donor and as a recipient. So what do we mean here? You must understand that a person of blood group A must always have antigen B. And as a donor, if you are a donor and your blood group A, you want to give out blood, you only give to A and AB alone, but you will not give to B and O. Why? Because the B and the O will always stimulate the production of anti-A, which will always come and fight the antigen on your surface of blood group. Actually, red blood cell surface. Excuse me. So, as a recipient, your blood group A, you are receiving. You will only receive blood from A and O alone. Why O is a universal donor? Yeah, because he lacks both. He lacks both the antigens A and B. So, you will not receive blood from B and AB because of what I told you. Yeah, that is the table that is containing of table. When your blood group B, your anti-B, as a donor, you only receive blood from B and AB. You will not receive blood from A and O. As a recipient, B and O will give you, but you will not receive blood from A and AB. So this is now AB, blood group AB. You neither have A or B antigens Are you together? And therefore, you only receive blood from AB alone as a recipient. Actually, as a donor, as you give out blood and your blood group AB, only give fellow AB, but you don't give any other type of blood group. Yet you are a universal recipient because you lack both the antigens, so you always receive from all the types of blood group. Why? Because you are a universal recipient and you lack both antigens. So let us look at blood group O. With blood group O, you must appreciate that a person of blood group O will always actually give blood to a person. As a donor, you will give blood to a fellow O person and all other person. Now, as a recipient, you only receive from O. Why? 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 Think. According to what I've explained, think. So, as we conclude up with the red blood cells, I would like to conclude it with actually showing you the latest type of blood group, which is called actually rhesus factor, or what we call the blood group D. Most of us call it blood group D. So it is that plus or minus you always see on people's blood groups. So a person can either be A plus, or A negative. Are you together? So we must appreciate that rhesus factor is actually very common these days in this process of actually finding blood group of different people. So when a person has got, for example, when a person has got a blood group A, and your surface is surrounded with antigens A. Then with the modern world, they've also found out that some people have got what we call actually rhesus positive. How do they become rhesus positive? That sometimes they will always find the anti-rhesus factor also surrounding their surfaces of red blood cells. Are we together? Therefore, so once they find that anti-rhesus factor surrounding it, so if your blood group A, we shall always add a plus. If you have that, if you lack it, you remain A negative. Are you together? So that is with the rhesus. That is with the rhesus factor. It is the modern type of blood group which is actually found just of recent, and you must appreciate that about 85% of the people have antigen of rhesus actually have antigens of this type, and the rhesus positive are rhesus positive and therefore don't make antigen bodies. So the remaining 15% actually have got rhesus antigens and they actually rhesus negative. So you must appreciate that rhesus negative is actually stimulated to make anti-rhesus bodies in conditions like pregnancy and blood transfusion, where we need you to go and get to know how does this come about. Allow me now tell you a few about the blood groups in summary, that when you go to the laboratory, it is very important for you actually, it is very important so that you know your blood group, because it can help you in terms of actually in case of accidents and we need to do blood transfusion. It saves time. You might not lose time actually. You might not lose life easily. So, and majorly in the laboratory, we use two types of methods to determine the blood group of a person. We either use what we call tile grouping, or what we call the tube grouping. And in these days, the most important method is what we call the tile grouping. Those who do medical lab know much about that. I will not go deep into that. What I will require you to do is to go and actually look at how they do it. Allow me talk little about the white blood cells, or what we call the leukocytes, and I tell you that we have 5 types of leukocytes. There are 5. and I must know that under normal conditions, the body has got less number of leukocytes in the body. Why? Because if you are normal, no need of leukocytes to be actually in large amounts in the body. Actually, I would actually phrase this statement and I say, under normal conditions, the person lacks white blood cells, but it is not right. You will always have a few because still you always have microbes in the body. So you must appreciate that these ones are only produced in the body in case of any engagement of microorganisms, and their main function is to carry out defense and immunity. On average, you have 4,000 to 11,000 white blood cells actually per liter of blood, and you must appreciate that these white blood cells are actually classified into two types. That is what we call the granulocytes. And under the granulocytes, those are white blood cells that actually have granules on them, and those are the neutrophils, the eosinophils, and the basophils. While they're also divided in what we call the granulocytes, which are actually called those cells that lack the granules on them. That is the lymphocytes and the monocytes. Let me actually look at the granulocytes. That is, those are the eosinophils, the basophils, and the neutrophils. And I must know that the eosinophils take up the red acid dye called eno. The basophils take up an alkaline methyl blue, and the neutrophils are purple because they take up both dyes. That is in the laboratory as they try to check for their presence. And you must appreciate that the neutrophils, their main function is to actually protect against foreign materials that gains entry into the body. They also remove waste materials, e.g., dead cells and dead tubules from dead tissues. Allow me to tell you that the neutrophils are attracted in large numbers in an area of an infection. Are we together? The neutrophils are highly mobile, and they can squeeze and pass through different capillaries. They have a lifespan of 6 to 9 hours. Every 6 and 9 hours, they always die. So, that is what I can always tell you, that they carry out the fighting of diseases by carrying out phagocytosis. Yeah, so that is what I can always tell you about the neutrophils. Allow me now proceed to the other types of granulocytes called eosinophils, and their major function is almost totally unknown, though they are normally comprised of 1 to 3% of all the leukocytes. They are also weak phagocytes compared to the neutrophils. Their main role is to eliminate parasites such as those ones I must have talked of in microbiology, the Ascaris lumbricoids and so other things. Let us talk about the basophils in brief, and I tell you that the basophils are actually comprise of only 0.4% saturating leukocytes, and their main function is actually help in fighting inflammation in the body by the help of the anti-inflammatory substances in the body, and they also help in allergy reactions. Those are with the basophils. As I told you first, the neutrophils, those ones fight microbes by phagocytosis. Eosinophils fight parasites. The basophils, the allergies and the inflammation. Now let us look at the granulocytes, which actually the monocytes or what we call the lymphocytes, and these ones play the same role of actually fighting diseases through what we call the cell-mediated type of or the cell-controlled immunity. You must have realized that the lymphocytes produced in the bone marrow produce for us the T lymphocytes, which T lymphocytes gave us the B lymphocytes. The B lymphocytes produced for us the antibodies that carried out killing of the microorganisms as phagocytosis takes place. Allow me look at the last cellular component of blood, which is the platelets, and these ones play a big important role in actually in a in a in a in what we call blood clotting. You must know that they are around 2 to 4 micrometers in diameter, and they are very many in the body during fighting during control of bleeding. Allow me tell you that the control of bleeding is actually a process called hemostasis. It's called hemostasis. It's the process of actual stoppage of hemorrhage or control of bleeding, and it is actually separated in actually what we call around 4 stages. That is the vasoconstriction, where we shall see thickening of the walls. The diameter will also reduce, and also no supply of blood will be sent to the tissue. It also consists of what we call the plate plug formation, and this one is where we shall see a wire mesh and the production of different chemicals to kill the microbes is done. We shall also see blood clotting, where we shall see both the fibrogen and what we call it is usually fibrinogen and some other thing I'm forgetting. Uh, it is actually fibrinogen and what is that? There is some other component that helps in and the prothrombin, and the prothrombin that will always come and help in actually in coagulation. Then we shall see thrombosis taking place, where we shall see wound healing actually taking place in the last phase. That is all I can always tell or talk about blood, and I would like to conclude by looking at process some of the disorders of blood that we have, and those include anemia, and this is the ability of blood, or this is the inability of blood to clot. We have what we call the leukemia. We have what we call the blood cancer, that is the same as the blood cancer. We have what we call the thrombocysis. We also have what we call the hemophysis, and so many other concepts. Martin Molondo is my name, and I'm happy to always share with you concepts concerning anatomy and physiology. Stay tuned, stay blessed, love you.